Vanakkam. Welcome to this course on Classics in Neuroscience. In this video, we will continue our discussion on neural circuits in the spinal cord, retina and invertebrate system. In this particular video, we will look at the pathways within the spinal cord identified by the work of Eccles and his colleagues, the Renshaw inhibition, and we will also discuss some perspective on Eccles and his legacy and his influence in the field of neurophysiology, neuroscience in general, neurophysiology in particular. As we have seen previously, Eccles and his collaborators continued their work on excitatory and inhibitory pathways within the spinal cord. What is shown here is the motor pathway exciting the muscle and the motor pathway that excites the antagonist, and the information that is taken by the sensory system. What happens when you perform the tendon tap experiment? You just tap the tendon, then what happens is that immediately there is an extension that is seen. So, that what happens, how is that working? Turns out that that pathway is a monosynaptic pathway to the 1A afferent. This reflex is also called as T reflex or the tendon tap reflex. Can also be elicited by electrical stimulation, in which case it is called as the H reflex. It turns out that this reflex, that T reflex, is a monosynaptic reflex. Exactly how it functions, I will keep it very brief. That what happens is that this muscle contracts, right, that causes stretch of this muscle, the antagonist. When the antagonist is sensing stretch, what it does, it is sensing stretch through the muscle spindles, and it through a monosynaptic pathway, the proprioceptor neuron, that is the muscle spindle neuron, that proprioceptor neuron is activating the alpha motor neurons that is connected to that muscle, thus causing contraction. In this case, what is happening, the tapping of the tendon causes a response that is massive, as if the hamstring muscle is contracting. Hamstring muscle is actually not contracting, but the sudden tap on the tendon causes the stretch receptors to become super active or hyperactive for a brief period of time, causing the activation of the muscle in a monosynaptic manner, right, through only one synapse. I will ask my students to share the video regarding how this monosynaptic reflex works, that is the monosynaptic reflex due to the 1A afferent. Please do share this, please do share the 1A interneuronal pathway and the 1B afferent, all the reflex pathways, excitation and inhibition within the spinal cord, all that set of videos my students will share with you. So, this is the monosynaptic excitatory pathway, but also what happens is when the muscle contracts, there is a capsule within the tendon that detects the tension within the muscle. This is called as the Golgi tendon organs. This is also a proprioceptor that detects force or tension produced in the muscle. This activates an interneuron that then inhibits an alpha motor neuron that is causing the contraction. Alpha motor neuron is causing the contraction. You want to keep the contraction of that alpha motor neuron within controllable levels. So, a form of negative feedback is given to that alpha motor neuron, but this is not monosynaptic, because it turns out that proprioceptor neurons only produce excitatory signals. So, they excite an interneuron, in this case called as the 1B interneuron, which then inhibits. This 1B interneuron is an inhibitory interneuron, it inhibits the alpha motor neuron. there is the disynaptic pathway from the Golgi tendon organ that inhibits the alpha motor neuron of the agonist, inhibition, but it also in a way disinhibits the antagonist through a not disynaptic but rather oligosynaptic pathway through the 1B interneuron and the 1A interneuron. Please do check these pathways from my Neuroscience of Human Movements course. When alpha motor neurons innervate and excite the muscle fibers, collaterals or branches from these alpha motor neurons also activate an interneuron, and this interneuron goes back to inhibit the alpha motor neuron and the alpha motor neuronal pool that causes this contraction or this excitation. So, that is, let us say this is an alpha motor neuron that is innervating a muscle, okay, is exciting this muscle. Then I am going to draw another neuron, that is inhibiting, black filled dot means inhibition, inhibiting this neuron. It turns out this neuron that is inhibiting that alpha motor neuron is excited by an axon collateral, is excited by this collateral, this branch of an axon. Why would you excite somebody who inhibits you? Why would you stimulate somebody who inhibits you? Because there is a need for this negative feedback to keep the amount of excitation in control. This black colored neuron that I have drawn is called as Renshaw cell. It is a form of recurrent inhibition. The inhibition provided by this neuron is a form of recurrent inhibition through this interneuron, that is the Renshaw cell. Single shocks in the ventral root of the spinal cord produced an action potential that travelled antidromically to the cell bodies. Then there was a form of long lasting hyperpolarization. Hyperpolarization means inhibition that actually lasted longer than what an action potential would last. So, long lasting hyperpolarization, inhibition. This was measured through intracellular recording, and this was due to the activation of interneurons that are activated by the collaterals of the motor neurons. This was long lasting, it was similar to the inhibitory postsynaptic potentials found in the alpha motor neurons. This cell was named as Renshaw cell in honor of the person of the medical scientist Birdsey Renshaw, who first predicted that this kind of long lasting hyperpolarization could be due to inhibitory interneurons that are getting activated by the axon collaterals. Was a prediction that Birdsey Renshaw made at Harvard. Unfortunately, Birdsey Renshaw also passed away due to polio, considered an irreparable loss for the field of neurophysiology. Of course, we have seen in the previous lectures that most synaptic integration happens in the dendrites. This was shown by David Lloyd and Wilfrid Rahl. Also, Wilfrid Rahl showed there are some sort of novel interactions that happen between dendrites in the olfactory bulb. This idea was strongly opposed by Eccles. Remember his paper, Wilfrid Rahl's paper, that originally suggested that the exponential decay was perhaps due to the activation or activity that you can see in dendrites. Was a paper that was submitted by Wilfrid Rahl in the 1950s, and this was opposed by Eccles, and the paper was rejected because Eccles said that this was probably due to some instrumentation problem, which was not correct. Initially Eccles opposed this, but later he invited Rall to co-chair a session on this topic in 1960s. And of course, in 1960s Eccles broadened his interest to other brain regions and spent the last 20-30 years of his life studying other nuclei such as cerebellum, hippocampus and the neocortex. At this time it is maybe useful to understand the perspective of Eccles the man. And this Eccles originally was born in Australia, then he went to Britain with an I, then he went to Britain, and from there he went to New Zealand. Then he went to, from there he also travelled as part of his work to USA, Switzerland, finally settled down in Australia, and took on significant teaching responsibilities, for example in Otago, when he was in Otago. Remember our previous videos where he taught a lot of classes in Otago, in Dunedin. So that helped him to ground himself in the fundamentals of neuroscience, and he mentored over 200 students and collaborators, and published 500 plus papers and several books. And the books were very influential in shaping modern neuroscience, thinking about modern neuroscience. He was a dynamic personality, he was very vocal. Remember his conversations with H.H. Dale about the soup versus Sparks debate. He was a very vocal and very dynamic person, and in the 1940s and 1950s Eccles' work contributed to improving our understanding of spinal cord reflex pathways, and then he shifted his interest to other fields. There is a detailed work, there is a detailed description in the book. Please do check the book for more details. It suffices to say that he was a complicated person. Many times he would first he said that transmission was purely electrical and not chemical, but later through his own work on inhibitory postsynaptic potential it was shown that there was indeed chemical transmission that happens. So as a scientist he kept on evolving, so to speak, but his personality sometimes may have hurt other scientists, may have been seen as harsh by other scientists. Do read up about this, I am not speaking further about this. With this we come to the end of this video. In this video we looked at the spinal cord pathways, the work of Eccles and his collaborators on the EPSPs and IPSPs, specifically on Renshaw's inhibition, and we also discussed about the personality of Jack Eccles. With this we come to the end of this video. Thank you for your attention.